Welcome to Biology at Priyojit, my dear students. In this class, we will understand prokaryotic transcription step by step, focusing on its three key stages: initiation, elongation, and termination. Transcription basically is the fundamental process by which genetic information encoded in DNA is copied into RNA, enabling protein synthesis in organisms like E. coli. By breaking this process into simple logical steps, we will see how RNA polymerase recognizes the promoter, synthesizes RNA, and finally stops transcription with precision and efficiency. So let's explore how prokaryotic cells control gene expression in a clear and exam-friendly approach. Overview. Transcription is fundamentally the synthesis of RNA from a DNA template. It is catalyzed by DNA-dependent RNA polymerase. It occurs in three stages: initiation, elongation, and termination. So this is the process of transcription where DNA is copied into RNA. So initiation involves the RNA polymerase binds to the promoter sequence of the double-stranded DNA and starts unwind the DNA strand. So what is the promoter sequence? Promoter sequence is the sequence of a DNA molecule where the RNA polymerase can bind to initiate the process of transcription. So this is the process of the fundamental of initiation and elongation. RNA polymerase when moves along the DNA template strand from 3' to 5' manner, and correspondingly the RNA is transcribed into 5' to 3' manner by addition of nucleotides to the 3' end of the growing RNA. So this is the summary of the elongation process. And when RNA polymerase reaches the termination site, the RNA transcript is set up from the template, that is the termination process, and the RNA has the same base sequence as the DNA coding strand but with U instead of T. We know that in RNA there is uracil instead of thymine. Thymine is present only in DNA molecule. Initiation of transcription, so binding of RNA polymerase to promoter. We have already studied about the RNA polymerase enzyme in our previous class. So in prokaryotes, transcription begins basically when RNA polymerase holoenzyme, that is coenzyme plus sigma factor, binds to the promoter region of DNA, and the promoter is located upstream or 5' site of the transcription start site. RNA polymerase then covers about 75 to 80 base pairs, extending from -55 to +20. So role of the sigma factor. Sigma factor is essential for promoter recognition and initiation. It recognizes -35 region, that is 5' TTGA 3' and -10 region, which is also known as Pribnow box, that is 5' TATA 83'. So optimal spacing between the -10 and -35 regions is 17 base pair, and the sigma factor also helps in the melting or unwinding process of DNA. Formation of closed and open complex. Closed complex. So what happens? RNA polymerase binds to double-stranded DNA at the core complex, and the open complex DNA melts between -11 to +2 positions, and it forms a transcription bubble. So this is the transcription bubble. It's a bubble like structure, Solin structure of about 11 to 12 base pair. It results a ternary complex. So why it is ternary? Because there is a DNA, RNA and protein. RNA polymerase, that is the protein component. So DNA is there, short RNA is there, and RNA polymerase protein is there. So it's a ternary complex. DNA melting and promoter recognition. We know that sigma factor regions are responsible for recognizing the promoter sequence. Sigma factor region 2.4 recognizes the -10 region. So this is the -10 region is recognized by the sigma factor 2.3 to 2.4, and region 4.2 recognizes the -35 region, and region 2.3, this is the 2.3 region again, they help in DNA melting. So aromatic amino acids of the sigma region 2 interact with the non-template strand to stabilize the melted DNA. Special promoter elements, we know that extended -tail element or 5' Tg3 prime that is present when minus 35 region is absent. So when minus 35 region is absent, there is an extended minus 10 element, that is 5 prime Tg3 prime, and it is recognized by sigma region 3. At its discriminator sequence, it is located downstream of minus 10 region, so it is recognized by sigma factor 1.2. Up element or UP element is located 20 base pair upstream of minus 35 region. It is recognized by CTD of alpha subunit. It is important for rRNA gene transcription. So initiation of RNA synthesis, we know that RNA synthesis starts at the +1 site. This is the +1 site of the RNA molecule, and usually it's a either A or G, that is it must be a purine base. So initial RNA synthesis is unstable, so there is an phenomena of abortive initiation. This is the image showing the abortive cycle. So what happens during abortive initiation? So this is the process, transcription initiation process in prokaryotes. This is the transcription start site. It is designated as +1. So alpha, the two alpha subunits, one beta prime, and one sigma and one omega subunits are there in the RNA polymerase enzyme, and it forms a transcription bubble. There is a Pribnow box or -10 base pair sequences where RNA polymerase binds, and this is the -35 region. They are also RNA polymerase binds. So this is the formation of the closed complex, and this is the formation of open complex when RNA polymerase binds the Pribnow box. So abortive initiation, what is abortive initiation? RNA polymerase often synthesizes shorter RNAs of 2 to 10 nucleotides long and release them. This process is called as abortive initiation. It occurs because RNA polymerase is still tightly bound to the promoter. Now we should study about the scrunching model. What is scrunching model? During early RNA synthesis, we know that trailing edge of RNA polymerase remains fixed and leading edge moves forward with each nucleotide added. So basically what happens, scrunching model explains how RNA polymerase starts transcription and escapes from the promoter during initiation in prokaryotes. So what scrunching model explain? When RNA polymerase starts transcription and escapes from the promoter during initiation in prokaryotes. What happens in the scrunching model? Firstly, during initiation, RNA polymerase binds tightly to the promoter. The trailing end of RNA polymerase remains fixed on the promoter, and the leading end of the RNA polymerase moves forward as the RNA synthesis begins. Number 4, for every nucleotide added to the growing RNA chain, the DNA is pulled into the enzyme, and ultimately this causes accumulation of unwound or single-stranded DNA inside the enzyme. So ultimately this causes this compressed expanded DNA enzyme structure is called the scrunched complex. So scrunching provides energy for promoter escape, release sigma factor or abortive RNA release. Promoter escape, what happens when RNA chain reaches approximately 10 nucleotide sequence, then RNA polymerase breaks promoter contacts. Sigma factor then dissociates and the stable elongation complex is formed. This marks the end of initiation. So this is the enzyme, this is the active site of the enzyme where RNA polymerase binds, and this is the direction of transcription. So elongation of transcription, what happens? The release of sigma factor indicates the elongation of transcription. Elongation begins after the sigma subunit dissociates. So core RNA polymerase continues transcription at that time, and RNA chain synthesis, RNA is synthesized in 5 prime to 3 prime direction. Rate of synthesis is approximately 40 nucleotides per second at 37 degrees Celsius, and incoming ribonucleotide they losses the beta and gamma phosphates. The growing RNA chain losses 3 prime OH hydrogen during the bond formation. So DNA unwinding and rewinding. DNA is basically unwound ahead of RNA polymerase and it rewound behind the enzyme. So RNA DNA hybrid alone is unstable, and RNA polymerase maintains the stability of the enzyme. So this is the image showing the elongation process of transcription. So transcription initiation and elongation in prokaryotes. So during initiation we have found that sigma factor is attached with the RNA polymerase enzyme, but when elongation is started, it is indicated by the release of the sigma factor and the mRNA starts to grow. We have found that what happens during elongation process, trinucleotide phosphate comes and one phosphate is added, and remaining beta and gamma phosphates are released. They are known as pyrophosphates, and from these pyrophosphates two inorganic phosphates or orthophosphates will be produced, which will required for energy purpose, and they will supply the energy to make the phosphodiester bond formation in the newly growing RNA chain. So supercoiling occurs during elongation. Movement of RNA polymerase causes positive supercoils ahead and negative supercoils behind. What happens? What enzymes are involved? We know that DNA guidage removes positive supercoil and topoisomerase removes negative supercoils. So proofreading by RNA polymerase performs proofreading by pyrophosphorylatic editing and hydrolytic editing. These ensure accuracy of transcription. So elongation factor, there are proteins involved, that is NAS-A, NAS-B, NAS-E and NAS-G. What are the functions? They regulate elongation rate, they participate in termination and anti-termination. What is the inhibitors of elongation? There is cordycepin, that is act as adenosine analog. They lack 3' OH group and they inhibit RNA chain elongation. So this is the barrier to further transcript elongation. Termination of transcription by termination signals instruct RNA polymerase to stop transcription and dissociate from the complex. There are two types of termination in prokaryotes. First one is the rho-dependent termination. The image here showing the termination, the newly synthesized RNA strand is released. So what is the role of rho factor? Rho is an ATP dependent helicase protein, and it is responsible for nearly 50% of terminations. RAT site binding, rho binds to a specific RNA sequence called RAT site, or also known as rho utilization site. RAT site is also located upstream of the termination site. Rho translocation using ATP hydrolysis, rho moves along the RNA towards RNA polymerase. So what is termination mechanism? When RNA polymerase pauses at termination sites, RNA catches up, it frees polymerase structure, it invades RNA exit channel, and it destabilizes the transcription complex. What is the result? Releases of RNA and dissociation of RNA polymerase from DNA. So this is the rho-independent termination process. What happens during rho-independent or intrinsic termination? There is an intrinsic sequence is there. So DNA template features, it contains GC-rich inverted repeat, which is followed by AT-rich region. Hairpin loop formation. Transcribed RNA folds into a stable GC-rich hairpin, and hairpin causes RNA polymerase to stall at that site. Weak RNA-DNA interaction following the hairpin. RNA contain many U residues. These are the U residues, and UA base pair with DNA is very much weak because there are only 2 double bonds, unlike the GC base pair where there are 3 hydrogen bonds. So release of RNA is combined effect of polymerase stalling and weak RNA-DNA bonding, and it causes dissociation of RNA polymerase and release of newly synthesized RNA. So this is the image showing the rho-independent termination process. This is also found in case of 50% genes. So completion of transcription. After termination, transcription is completed. In prokaryotes, transcription and translation occur simultaneously, so it is called as co-transcriptional translation, and there is no nuclear membrane, so mRNA can be translated even before transcription ends. So transcription, translation and mRNA degradation are all coupled and proceed in 5' to 3' direction. So this is the image showing the intrinsic transcription termination. So as we conclude that, remember that prokaryotic transcription is a beautifully coordinated process from promoter recognition by the RNA polymerase during initiation, precise RNA synthesis during elongation, and to accurate stopping at termination. Each step is tightly regulated by RNA polymerase. Understanding these stages not only strengthens your concepts for undergraduate, postgraduate and NEET exam, but also helps you appreciate how cells efficiently convert DNA information into functional RNA. Keep revising, stay curious and continue exploring the wonders of molecular biology. Thank you.